Fusarin is a pigment first isolated in 1937 by Julius Nicholson Ashley from the fungal plant pathogen Fusarium clamorum. Today, however, it's known as a potential anti-cancer agent. The Porco group at Boston University in 2018 published a paper describing the synthesis of the compound. However, while the compound can be synthesized successfully by these methods, there's a need to reduce the number of steps. So the purpose of my project was to determine a more efficient synthesis of Fusarin. So we were able to perform the first three steps of the synthesis, the first being a methylation reaction in which a methyl group, or CH3, was added to a substance called 2,4,5-trimethoxybenzoic acid. This product that was formed was called an ester. The second reaction involved using that ester as the reactant. The ester was oxidized by a substance called cearic ammonium nitrate, abbreviated to CAN. In other words, the CAN removed electrons from the ester, and the product that was formed was called a benzylquinone. The third reaction is called a Michael addition reaction, in which a substance called a pyrone was added to the benzylquinone from the previous reaction. So what we found out was that the methylation reaction, the first reaction, was easily reproducible, high yielding, and we were able to get very pure product. And even when we increased the scale of the reaction from 200 milligrams to a whole gram, that's 5 times the amount, the yield only decreased by 8%. For the second reaction, the CAN reaction, it wasn't as reproducible as the first one, as things in chemistry generally are not. In the CAN reaction, sometimes the reactions didn't even go to completion. But we were able to figure out that by switching to using round-bottomed flasks, which have more surface area compared to a small vial that we were using before, we were able to achieve better stirring, which then prompted the reaction to go to completion more often. Also, when we increased the amount of CAN in the reaction itself, the reaction also went to completion more often. The third reaction, the Michael addition reaction, we were only able to do once, and we had limited success with it, most likely due to the fact that for this reaction, the reactant was a product from the last reaction, and since that wasn't especially pure, the product for this reaction also wasn't as pure as it could be. So in the future, we'd definitely like to reproduce especially the second and third reactions, or the CAN oxidation and Michael addition reactions, as well as continue in the general synthesis of Fusarin. The synthesis of Fusarin can also be extended to studying the compound diaporine, which can serve as a barrier to the process which converts macrophages, or white blood cells, from repairing cells to killing cells.